Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 3.2, environmental impacts on enzyme function. Objectives today are to explain how changes to the structure of an enzyme may affect its function, and we'll be looking specifically at environmental changes, namely denaturation due to temperature or pH. We'll also be looking at competitive and non-competitive inhibition as a result of the environment. And suggested skill will be argumentation 6E, predict the causes or effects of a change in or disruption to one or more components in a biological system. First up, temperature and pH. As was mentioned previously in 3.1 and 3.2, enzymes are, for the most part, proteins. This means that they're going to follow the protein folding rules of proteins, and they're going to have primary, secondary, tertiary, and quaternary structure. And as we learned back in Unit 1, changes in pH or temperature can destroy the weak bonds of tertiary and quaternary structure. And this can cause the enzyme to denature. Now, each enzyme has an optimal zone for both temperature and pH. If we look here, there's an optimal temperature for two enzymes, optimal temperature for a typical human enzyme, and an optimal temperature for heat-tolerant bacteria. Because the heat-tolerant bacteria is heat-tolerant, the enzymes within it are able to work at a higher temperature, and that is their optimal range, as you can see, compared to the human enzyme. However, the reason for why there is a sort of steep incline in terms of reaching this optimal range is because generally as heat and temperature is increased, molecules are going to be moving around faster, and there's a higher chance of substrates entering active sites at a faster rate if molecules are moving faster and bumping more into each other. So that's why you see as you increase temperature, even though you haven't quite reached optimal ranges yet, there's still going to be a steady increase of rate of production and reaction because just more substrates are hitting active sites. However, once you go past this optimal range, there's pretty quick drop in production because the enzyme will denature from too high of a temperature. Similarly, there are going to be different optimal pHs for enzymes depending on where they're located in the body and just what their function is. And here you can see there's a clear optimal range, and the enzyme is going to work outside of this range, but not by much. And this brings me to my next point, which is compartmentalization. Often enzymes are within certain enclosed membrane spaces because of their specific pH and temperature requirements, such as within the lysosome. They're hydrolytic enzymes that function at an extremely acidic pH, and they're located within the lysosome because they would not be able to function outside of that environment. And sometimes denaturation is reversible. If an enzyme is denatured through too high, too low temperature or pH, it is then able to sometimes be put back together through the tertiary bonding. Okay, so now we have to take a look at some graphs and make predictions. The graph we're looking at is product over time, and as you can see, in the beginning, there's substrate filling up the active site of the enzymes, and eventually all of the substrate has been used up, and we don't see any more increase of product over time. Now we have to ask ourselves, how would the graph change if enzyme concentration increased? So that means you're going to have more enzymes for the same amount of substrate, more initial active sites that can be filled up. So all of your substrate is going to be used up faster. This means that there's going to be a steeper curve here, and you're able to get to this point of leveling out much faster because you have more enzymes to use up that same amount of substrate. Now, what would happen if the substrate concentration is increased for the same amount of enzymes? This means you don't have more enzymes, but you do have more substrate. So it's going to be longer before all of the substrate is used up, and the graph levels out. So this graph is going to look a little bit like this. It's going to still be increasing, and even when this graph levels out, this one is going to continue increasing, and it's going to level out a little bit past the other one. Now, besides the enzyme and the substrate, there are a couple of other factors that we have to look at play here. First of all are cofactors, and these are non-protein helpers that bind to the active site and assist in the reaction. These are metal ions such as zinc, iron, and copper. And a type of cofactor that is organic is known as a coenzyme, and these are like vitamins. As you can see, they bind to the active site of the enzyme, and the substrate would not have been able to bind to the active site had there not been this cofactor or coenzyme. The hexagon would not have fit into the circular shape here, but it only does so after the cofactor or coenzyme has been bound to the active site. We also have to talk about inhibition, and there are two kinds of inhibitors. First of all, there are competitive inhibitors, and these are molecules that are competing with the substrate for the active site on the enzyme, and these can be irreversible. Essentially, they're blocking the enzyme function indefinitely, and poisons are one type of competitive inhibitor. They usually have the same shape as substrates and can reduce reaction efficiency because they're using up this enzyme and they're not letting go, which essentially prevents potentially very important reactions from occurring in your body. So over here we have a competitive inhibitor. This is the inhibitor here that's within the enzyme, and it's not letting the substrate bind to the enzyme, which is stopping the reaction from happening. It's inhibiting the reaction. Now, there are also non-competitive inhibitors, and these are molecules that impede with enzyme activity by binding to another part of the enzyme, called the allosteric site. So non-competitive inhibitors, they don't stop the substrate from entering the enzyme, but the problem is that they cause a change in the shape of the enzyme, so the enzyme is no longer the right shape to accept the substrate. And non-competitive inhibition is a way to regulate enzymes. So substances that bind to the allosteric site can both inhibit and stimulate enzyme activity through the shape change it causes. For instance, if an inhibitor binds to the allosteric site, that's going to prevent the enzyme from being able to accept the substrate. And this is going to inhibit enzyme activity. However, there are other molecules that can bind to the allosteric site, which allow the enzyme to then be changed into the right shape to accept the substrate, which stimulates enzyme activity. Now, feedback inhibition is when the end product of an enzymatic pathway switches off the pathway by binding to the allosteric site of an enzyme in the pathway. And this is going to turn off the pathway when enough of the product is present in the cell. And this helps increase efficiency because you're not making surplus product that you don't need. So within this enzymatic pathway, where C is going to be the final product, C can then be the inhibitor for enzyme 1, and C can bind to the allosteric site of enzyme 1, causing a shape change which prevents enzyme 1 from turning reactant A into B, which is then going to turn off this whole enzymatic pathway. And C is only going to do this once the cell realizes that, hey, we've got enough of you in here, we don't need any more, and that way the cell is able to then save energy by not making more than what it needs. This has been a pretty packed lesson, but here's some environmental impacts on enzyme function. So, enzyme denaturation and optimal ranges. pH and temperature can both denature the enzyme when they're too low or too high, stopping enzyme function. There are effects of environmental temperature and concentration changes on enzyme activity. As temperature increases, generally enzyme activity is going to increase as well as molecules are moving around faster until you go past this optimal zone where then the enzyme denatures. And obviously, if you have more enzymes or substrate, that is going to change the speed of reactions over time, just like we looked at with the graph. And lastly, enzymes are regulated by the presence of inhibitors, both competitive and non-competitive.